In this section on the endocrine system chapter, we're going to be looking at hormones. We'll consider how to classify them, their chemical natures, how they travel through the bloodstream, and how they interact with target tissues and interact with the receptor systems. First, in considering the signaling molecules that are hormones, we want to think about how they're secreted. Now, many of the hormones that we're going to be considering are protein hormones, which we'll talk about soon, although there are other types of hormones. This picture, which comes from the chapter on cell biology in the textbook, is looking at essentially the secretory pathway. So in the nucleus, there's going to be a gene, say the insulin gene, which gets transcribed, and the mRNA moves out of the nucleus and into the cytoplasm. For secretion specifically, it's going to go to the rough ER. That ER is rough because it has ribosomes on it. Those ribosomes will translate the mRNA into a protein, and in doing that, it ends up in the lumen of the endoplasmic reticulum. It'll move through the endoplasmic reticulum's internal space towards the edge where it ends up being packaged into a transport vesicle, which will move to the Golgi apparatus. The receiving side of the Golgi apparatus is called the cis phase, and then the protein will move from layer to layer of the Golgi apparatus until it reaches the trans phase, where it ends up being packaged into a secretory vesicle. In the Golgi apparatus, many secreted proteins are processed a little bit further so that they're ready for secretion. And then the secretory vesicle will merge with the plasma membrane and release its contents into the extracellular space. Now, in this picture, again, from just the cell biology chapter, what it's representing is a membrane-bound protein, which might be a receptor protein or something to that effect. But really for hormones, it's going to be releasing that molecule into the space. It won't remain attached to the cell membrane. When we talk about the endocrine system and hormones, we are usually imagining signaling molecules that move from the source, the gland, into the bloodstream, travel throughout the body actually, and then finally meet up with their target tissues where the hormone binds to a receptor. Without a receptor, hormones have no effect on a cell. It's only when they bind to the receptor, which the cell expresses, that there's a target reaction. There are two other aspects of signaling which can't really be separated from the endocrine system, and those are paracrine and autocrine. Paracrine refers to signaling that takes place between neighboring cells, so there's no intervening bloodstream. And then autocrine is when a cell signals itself. Now, especially the autocrine situation might seem strange, but there are certainly examples of this. A good example actually is from the stomach. There's a cell called a G cell which secretes the hormone gastrin. Gastrin will activate the G cell itself, stimulating more gastrin production. Gastrin will leave from the G cell and activate other cells in the gland, which are responsible for making the gastric juices, acid and enzymes and that sort of thing, so that it'll be more active. That would be autocrine and paracrine effects of that particular hormone. Gastrin can also get into the bloodstream where it'll have an effect on other aspects of the stomach or possibly the intestines. So it can really operate in all three realms. To separate them out really isn't entirely meaningful because these molecules could do any of the three possible things mentioned here. All that's important is that the target tissue has the receptor for that signaling molecule. And sometimes a cell has a receptor for its own signaling molecule, and therefore it interacts with itself. In considering the hormones, we want to think about how to classify them. Now, the book begins by classifying hormones based on what type of molecule they are. And this is important. It defines them chemically, certainly, but there's another aspect to classification that I want to touch on in a little while. First are amine hormones. These are amino acids that have been modified to become particular types of signaling molecules. The example given here is norepinephrine, which is modified from tyrosine, and tyrosine as an amino acid is characterized by the six-sided ring structure that we see here. Norepinephrine is just slightly modified from that. Norepinephrine is actually part of a slightly larger group, which includes epinephrine and dopamine as well, which can all be signaling molecules in their own right. Epinephrine is most classically considered a hormone from the adrenal medulla, where it has a part to play in the fight or flight response. Norepinephrine is also released there, just in far less quantities, but norepinephrine is more well known as a player in the sympathetic projections. The postganglionic fibers release norepinephrine onto target cells. Dopamine is used primarily as a neurotransmitter also, but in more somatic nervous system roles. A special example of an amino acid derived hormone, or two of them actually, are the thyroid hormones, T3 and T4. Now, you can see in these various versions of the hormones that there is the same six-sided ring structure that we saw in norepinephrine, which, like I said, comes from tyrosine. However, we see two of those ring structures. Now, T4 we see at the top of this figure and has the amino acid structure still incorporated into it. The carboxyl group COOH and the amino group NH2. That aspect of the molecules are still present in the T3 variations in the rest of this figure, but they're represented just by the letter R, so that they don't have to just keep being rewritten and make this an even busier figure. But what we do see are the two tyrosine ring structures. There is an important difference between the amino acid type hormones like norepinephrine or epinephrine and the thyroid hormones. These two ring structures in the thyroid hormones actually make these molecules quite hydrophobic. And we'll talk about the chemical properties of hormones in a little while, but keep that in mind. The next two types of hormones are demonstrated together here because they're actually fairly similar. Those are peptide hormones and protein hormones. And the difference between those is just how many amino acids there are in the chain that makes up the molecule. Peptide hormones are short chains and proteins are long chains. Now, proteins are usually a specific gene product. There's a gene for example, growth hormone. Whereas sometimes peptide hormones are a fragment of a gene product. And it might be that several hormones of a similar class are all translated from one gene, and then that gene product is broken down into the individual pieces that are then going to be individual peptide hormones. But otherwise, they are fairly similar. They're just chains of amino acids. There'll be three-dimensional shapes associated with that. The picture of oxytocin here is a two-dimensional representation of the chain, also pointing out that the two cysteine amino acids get linked together, making a loop structure in there. It does have a three-dimensional shape that's just not apparent from this. And then the growth hormone picture below that shows a ribbon model suggesting the three-dimensional shape of that hormone. Most of the hormones that we're going to be considering in this class are going to be protein hormones, actually. So that'll be the most common aspect of them. The last type listed here is the steroid hormone, which is characterized as being derived from the lipid molecule cholesterol. Now, cholesterol is most easily recognized by its chemical structure of the four-ring structure that we see in the two examples, testosterone and progesterone. What's different about cholesterol is exactly what's attached to those ring structures. For example, the two carbons that are attached to the five-sided ring structure in progesterone are part of a longer carbon chain that would be present in cholesterol. That's just been cut down, so to speak, to give us the steroid hormone. And then testosterone doesn't even have carbons in those positions. It's just a hydroxyl group there. Now, other steroid hormones would include the estrogen hormones like estradiol, or the corticoids, which are coming from the adrenal cortex, like cortisol and similar molecules there. But they are collectively a class all derived from the lipid cholesterol that we get in our diet. Besides the chemical nature of the source of those hormone molecules, it's also important to consider their chemical properties and how they do their job as a hormone. And in that means I prefer to consider hormones into two major groups, which are lipid-soluble hormones and water-soluble hormones. Lipid-soluble hormones are going to be mostly hydrocarbons and therefore nonpolar or hydrophobic, and water-soluble hormones are going to be polar or hydrophilic. Of the groups that we just looked at, most amino hormones or amine hormones are going to be water-soluble, with the exception of the thyroid hormones T3 and T4. The double rings from tyrosine make the thyroid hormones much more nonpolar and therefore lipid-soluble. The water-soluble hormones also include the peptide and protein hormones, as proteins tend to be water-soluble. And then the lipid-soluble hormones will include the steroids that we just saw. Another type of hormone which we won't consider in this chapter, but I do want to mention for a second, are the eicosanoids, which are a class of hormone mainly used by white blood cells. And so we will talk about those a little bit in the immune system chapter. And in that, I want to concentrate on prostaglandins as one of those types, and we'll talk about prostaglandins in the immune system chapter. There are two important aspects to consider when we're comparing lipid-soluble and water-soluble hormones. The first of which is transport. Hormones, as I said earlier, are mainly traveling through the bloodstream, and the bloodstream, of course, is a water environment. Plasma is mostly water. And so it's important to consider how these types of hormones move through the system. Now, water-soluble hormones, being water-soluble, are going to dissolve in the blood just fine. The lipid-soluble hormones, however, have to associate with special proteins that can help them dissolve into the water of the plasma. Now, for a lot of lipid-soluble hormones, they use a plasma protein called albumin. And albumin has its own role to play in blood, which we'll talk about in the next section when we start the cardiovascular system. But it's also has pockets where lipid-soluble molecules can slip in to be protected from the water environment of the blood. The thyroid hormones, T3 and T4, also have dedicated transport proteins for them, which are also made in the liver along with albumin, but have different affinities for those molecules. There's one that has affinity for both T3 and T4, another that has primarily affinity for T4. The specifics aren't important, but there are special transport proteins to carry those through the plasma. The other important aspect of considering the solubility of hormones is how they react with their receptors. Now remember, receptors are necessary to call a cell a particular target of a hormone. Without a receptor, the cell will not respond to the hormone whatsoever. The difference between the hormone receptors has to do with the solubility, in that lipid-soluble hormones can pass through the lipid environment of the cell membrane, and so those receptors are found inside the cell. Water-soluble hormones, however, since they can't cross the cell membrane, are going to bind with receptors on the surface of the cell, transmembrane proteins. Now, the types of receptors that you're familiar with probably from looking at the nervous system are really that type, that transmembrane receptor sort. However, there's a slight difference in how they're used in the nervous and endocrine systems. We're going to look at a specific water-soluble hormone receptor for hormones, which is the rule for water-soluble hormone receptors. In the nervous system, the same type is used, but there's another type that's used also, which you studied when you learned about the neuromuscular junction and when you were talking about the nervous system in general. These two figures represent the ways that lipid-soluble or water-soluble hormones interact with their receptors. In this picture representing lipid-soluble hormones, there is one thing to point out that's a little incorrect. The lipid-soluble hormone, represented by the yellow oval in the capillary, would of course be associated with the transport protein. But that's not a crucial part of this picture, just to understand that that would be true. The hormone, after it leaves the bloodstream, would move to the cell and actually cross the cell membrane easily, being a lipid. And it will react with or complex with the receptor in the cell itself. It might be taking place in the nucleus or in the cytoplasm. It sort of depends on the type of hormone, but that's what we'd see. And then it will move most likely into the nucleus, where it binds with the DNA and directly affects the transcription of genes, and therefore the translation of proteins as a result. Now, what we see here is probably a great representation of an anabolic steroid such as testosterone, which would get into a muscle cell, for instance, and direct the transcription of muscle proteins to increase muscle mass. But just as easily, this could be a thyroid hormone getting in and affecting metabolic enzymes. And extending the thyroid hormone idea, it could also get into the cytoplasm, complex with the receptor, go to a mitochondrion, and direct carbohydrate metabolism directly in the mitochondrion. So there are a number of things they can do, but they're all similar in that the receptor hormone complex is a special structure that interacts with components of the cell. In this picture, the water-soluble hormone, represented by the yellow triangle in the capillary, is of course going to be traveling alone. It doesn't need any help transporting through the bloodstream. I do find it amusing in this picture that the protein is represented in this picture like a pimple or something on the surface of this cell. It would in fact be a transmembrane protein. Just the detail on this picture of the cell isn't representing that so much. Now, the water-soluble system is a little bit specialized. The hormone binds to a receptor protein in the cell surface. And then that activates a G protein, which hydrolyzes GTP, causing it to physically move from the intracellular side of the receptor protein to an enzyme, usually generally called an effector protein. In this example, there's a specific name for it. That effector protein will generate a signal within the cell called the second messenger or secondary messenger. The secondary messenger will then go on to cause metabolic changes within the cell of different sorts. In this example, again, it's a specific example, it's activating a particular type of enzyme called a kinase, and then the kinase goes and adds phosphate groups to other proteins, activating them. The specific effector protein here is called adenylyl cyclase. Some sources will refer to it as adenylate cyclase, which, using the hydrolysis capacity of the G protein, will hydrolyze ATP to AMP, and then adenylyl cyclase cyclates AMP, which is just to say it makes the molecule into a circular or cyclical structure. That structure is not terribly important, but it just happens to be what that particular molecule is. And then CAMP, cyclic AMP, will go throughout the cell and have effects. It's called a second messenger because the hormone would be the first messenger, and that first messenger can't get inside the cell, so the second messenger is carrying out functions inside the cell. Other examples of second messengers can have other effects besides what's suggested here. It can also have an effect on the nucleus and gene transcription, that sort of thing. So there's a lot of variety in what can happen here. The adenylyl cyclase example is used because it's probably the most common type of effector protein in these sorts of receptor complexes. This figure does a good job of giving a very general impression of what this water-soluble hormone type receptor is. Now, the receptor is referred to as a Gproteincoupled receptor, or GPCR as the abbreviation. And again, it's not strictly for hormones. It's used in the nervous system as well. The agonist, or the molecule that binds to it, which would be a hormone for the endocrine system or a neurotransmitter for the nervous system, fits into the receptor protein that's in the cell membrane and activates the G protein, which then physically moves over to activate the effector protein, which will then make the second messenger, which causes the cellular response inside the cell, since the agonist, or the hormone in our case, can't get inside the cell. So really the G protein coupled receptor has three main components: the receptor protein, the G protein, and the effector protein. The second messenger is not part of the receptor, it's the result of what the receptor does. It's just taking the first message and translating it to a second message inside the cell. Now, consider the question, where are the proteins made that transport the lipid-soluble hormones in the plasma? The answer is the liver. albumin is the main transport protein that we find, and it is certainly made in the liver. But then also the special transport proteins that are used by the thyroid hormones are also made in the liver.